Good day, I am Canon Iram C. Constantino from 1D-BSN. So in this presentation, I'm going to talk about my understanding of the physiology behind muscle contraction. So for a movement to happen, the muscles in our body need to contract. Muscle contraction is the basis of all skeletal movements. The muscles in our body contract in a process wherein the nervous system, the skeletal system, and the muscular system interact with one another. A muscle contraction happens when muscle fibers regenerate tension through the movement of actin and myosin, which I will explain later. So, our muscles have several purposes in our body. For instance, when our muscles contract, it helps us maintain our posture whenever we are standing or sitting. Another thing is that it offers stability to our joints and connective tissues, such as our muscles tend to lengthen and shorten involuntarily as our body needs them. The skeletal muscle contains muscle fibers which are made of functional units that we call the sarcomere. This sarcomere contains two main muscle filaments, which are the thin filaments called actin and the thick filaments called myosin. The cross-bridge cycling theory is what forms the molecular basis for this so-called sliding movement. So when the actin and myosin overlap with one another, it will result in the shortening of the sarcomere in the muscle. This process is known as the sliding filament theory, which is also known as the most popular theory regarding this topic. So now let me talk about the cross-bridge cycle. It has 5 steps. So for the first step, the troponin units on the actin myofilaments are bound by calcium ions. The binding displaces the tropomyosin along with the myofilaments, which then exposes the myosin binding sites. For the next step, the head of each myosin units and the phosphate molecule remaining from the previous muscular contraction is now bound to an ATP. For the third step, the myosin heads will now release the phosphates and bind to the actin filaments with the newly exposed myosin binding sites. For the fourth step, the two myofilaments will then glide past each other and are propelled by the head-first movement of the myosin units, which is powered by the energy stored in their head. So the gliding motion will only be stopped only when the ATP molecules bind to the myosin heads, which severs the bond between myosin and actin. So for the last step, the ATP molecules will then be decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads. And now it is prepared to be used in the next cycle of movement. The myosin heads will now resume their starting positions along with the actin myofilaments and can now begin a new sequence of actin binding. And with that, the presence of further calcium ions will trigger a new cycle. And lastly, relaxation. In relaxation, the motor end is no longer stimulated. The calcium ions are now back into the sarcoplasmic reticulum, and the linkage between the actin and myosin is now broken. Thus, we can say that the muscle fiber is now relaxed. So, I have come to an end in my presentation, and these are my references. So, that's all. Thank you.